Okay. Apologies for the technical issues. Can everyone hear me? Okay, fantastic. Thank you so much for inviting me. It is an absolute pleasure to be talking to you today. I sincerely apologize if there are any more technical issues, but yeah, we'll try and do as best as we can. So what I'll do just to make sure that, so on my current screen, I can't actually see you. So what I'm going to do is every probably couple of minutes, I'm just going to check that I'm still talking to you. So apologies for that. Again, thank you so much for inviting me. It's been an absolute pleasure. It was an absolute pleasure when Ali. Much for inviting me. It's been an absolute pleasure. It was an absolute pleasure when Ali. Getting a lot of feedback. Ali. Okay, well, I'll start talking, and hopefully the issues will go away, the gremlins, and will let me go. So thank you so much for inviting me. That was a very nice introduction from Ali. About maybe 20 years ago, Matthew Rushworth went to give a talk in Tehran and told me of all the amazing things and people that he spoke to when he was there. So it's a real honor to be able to do the same as him. Yeah, so my group is very focused on decision making. And specifically, what we're interested in is how we weigh up the different costs and benefits of different decisions and different choices before we make an action. And so, kind of a nice example of that type of situation is this kind of situation here, where you've got two small primates who have surveyed their environment, and between this ice cream and this balloon, they've clearly decided that the ice cream is the best option in their environment. This picture kind of nicely sums up a number of things. The first is that human primates and non-human primates have a kind of similar idea about what's valuable in their environment. And so what I'm going to do is I'm going to spend most of my talk today talking about how these non-human primates, rhesus macaques, make decisions as a way of trying to understand how these non-human primates, these human primates, make decisions. And so, as Ali was saying, I was trained by Matthew Rushworth, who's an experimental psychologist. And so when we're trying to understand the decision-making process, what we want to do is break it down into its constituent parts. And so if we want to kind of think about how this monkey is making his choice, well, first of all, he has to identify the different options available to him. He then has to evaluate those options. So he has to kind of weigh up the benefits of the ice cream against the cost of having to retrieve the ice cream from a screaming child versus the other option. So the other option is obviously a balloon, has some aesthetic qualities. Maybe he'd think he could show it to his friends and they would think he was cool. And the cost is really just going over and retrieving the balloon. So once he's made his choice, he can then go to retrieve the ice cream. He can anticipate, he can start to think about how wonderful it would be when he gets to eat it. And then when he actually gets to eat it, he can then decide, well, was that worth it? Was it worth the investment and the cost of having to retrieve the ice cream? And then he can actually reevaluate and think and learn and think, okay, so would it be worth doing that in future? And this is the type of decision that we make every single day in our daily lives. Maybe not, we don't have to steal things from children, but instead we actually have to decide on what we want to do, where we want to spend our time, and the things that we want to pursue. So one of the things that my group has been really interested in is one kind of specific problem in this type of decision making, where really what's going on when the monkey is making this choice is he's weighing up the benefits, so the calories and the identity of this ice cream and what it's worth to him against his probability of success. That's how likely is he to actually be able to retrieve that ice cream from the child versus the qualities of the balloon versus the kind of complete certainty that he can probably retrieve the balloon. Maybe the balloon could float away from him, but he's pretty likely to be able to go and get it. So this type of problem, this integration between the benefit of an outcome and the probability of success is actually a problem that's been thought about for a long time. So people like Pascal and de Fermat over 200 years ago actually thought about and talked about this problem between the two of them, and they came up with a way of kind of thinking about it, which they called expected value or utility. And really, very simply, all this is it's the probability of success multiplied by the quality of or value of the thing that you're trying to choose on. And this has had a really kind of wide-ranging impact in neuroscience and also and most notably in economics, where it's really kind of formed the basis of kind of much of kind of microeconomic thinking. So I got really interested in this type of decision making and kind of valuation mechanism. And so when I went to work with Betsy Murray, one of the kind of the big ideas was that the orbitofrontal cortex in the frontal lobe must be one of the areas that's really, really important for this expected value computation. And the reason for thinking this is that was based on a number of experiments that Betsy had done with Alicia Izquierdo and a few other people around that time. And what they've found is that lesions to orbital prefrontal cortex, so on the ventral surface of the frontal lobe, caused deficits in both reversal learning, and this is kind of the classic effect that people have found many, many times, which is that when you lesion orbitofrontal cortex, animals have a problem reversing the contingencies when the contingencies of reward reverse between two different options. Again, this type of effect has been found in many, many different species, from humans to monkeys to cats to rats to mice and many others. And what they also found, so that kind of reward contingency is kind of like the probability side of the expected value equation. So the other side is then this kind of valuation side of the expected value equation. And Betsy and others had also found that aspiration lesions of the orbital cortex cause a deficit in this task called the reinforcer devaluation task. And so the idea here is that these are the two components of the expected value equation. So this was great. They had this strong evidence that it was involved, the orbital cortex was involved in both. But when I went to work with Betsy, we wondered if this was really true, and actually it turned out not to be. So in a series of studies, we looked at, instead of aspiration lesions, which damage fibers of passage near to the lesion site, we looked at how excitotoxic lesions would alter animals' behavior, and excitotoxic OFC lesions, I should say. And what we found is that the orbital cortex, even though it was completely destroyed using an excitotoxin, didn't seem to have any role in reward contingency. At least it didn't seem to, at least in the task that we were using. Instead, it seemed to be much more important for this valuation function as kind of assessed by the reinforcer devaluation task. So this led us to think, okay, maybe actually what's going on here is that the OFC is actually really only important for one part of the expected value equation. It's really only important for this valuation part, but not for the kind of this representation of probability or contingency. So we started wondering, okay, where might that type of process be happening? So this difference in lesion method between excitotoxic and aspiration lesions indicates that we must be causing a disconnection of another part of the brain that would be solving the kind of probability side of the expected value equation, which then leads to the question, okay, where? And so one of our strong hypotheses was that what we were doing when you make an aspiration lesion of the orbital cortex is that you're damaging fibers of white matter fibers coming from the temporal lobe, such as the uncinate fascicle. And so our thought was, what's really happening here when you make an aspiration lesion of the OFC is you're damaging the orbital cortex, but you're also blocking these fibers getting to other parts of the frontal lobe. The question is then, which part of the frontal lobe? So we knew that medial lesions of the medial frontal cortex don't cause deficits in this type of contingency learning. So our thought was that maybe it's this kind of more lateral, ventrolateral region of the frontal cortex that must be really important. So to test that, we compared monkeys with lesions of the ventrolateral frontal cortex, so that's Walker's areas 12 and parts of 45 on the lateral surface, against animals that had excitotoxic lesions of the OFC. And what we found using a probabilistic learning task was that animals with ventrolateral frontal cortex lesions were densely impaired at learning probabilistic associations. So again, that really suggests that that part of the brain must be really important for the kind of the probability side of the expected value equation. By contrast, lesions of that area did not in any way affect reinforcer devaluation performance. So we had this nice double dissociation suggesting that the ventrolateral frontal cortex is important for the kind of the probability side of the expected value equation, whereas the orbital cortex is really important for the valuation side of the equation. And that putting these interaction between these two areas is then important for actually computing the expected value of different options. But of course, when we interpret the effects of lesions, it's really important to remember that it's not that I and others think that this is that is the only kind of this area is the only area that's computing this type of process. It must be doing it within a network of areas that are interacting and kind of interacting to kind of produce these effects. So what I'm going to tell you about today is a series of experiments where we've really gone to try and probe the network and kind of circuit level function of different pathways and different mechanisms in the brain that might be important for this type of computation and decision making that's really, really important for kind of weighing up the different costs and benefits as we go about our daily lives. So I'm just going to check that I can still see you. You can still see me. Okay, I think we're still good. Okay, great. Okay, so I'm going to tell you about 3 different projects that we've run in my lab at Mount Sinai, conducted by different members of my lab, really getting at these, getting at different questions. So the first set of experiments I'm going to tell you about were conducted by Megan Fredericks, who was also at the NIH at the same time as Ali and I, who then came to do a PhD with me. And so what Megan and I got really interested in is wondering about how different pathways that connect from parts of the amygdala and limbic system to ventrolateral frontal cortex might be involved in probabilistic learning. So as I just told you a moment ago, we knew that the ventrolateral frontal cortex was really important when monkeys had to learn and use probabilistic reward associations to guide their behavior. So lesions of this area caused a dense impairment of monkeys' ability to learn probabilistic associations in this task that we'd used. But we also knew, actually, from work that had been done by a colleague at NIH, was that what we also knew was that lesions of the amygdala also caused a deficit in this type of learning. And actually, this experiment's really interesting. Should you care? They actually showed really quite nicely that ventral striatal lesions don't cause quite the same effect, despite the fact that numerous neuroimaging studies have very strongly indicated that it should. So what Megan and I started to wonder is, okay, maybe it's this interaction between the amygdala and ventrolateral frontal cortex that's important for probabilistic learning. And so what you're seeing here, this is just showing that there's a nice strong connection between amygdala and the ventrolateral frontal cortex. So that's area 12O, seen here from work by Helen Barbas. And so what you're looking at is fibers coming up from the amygdala to the ventrolateral frontal cortex are in green, whereas these kind of red and blue dots are actually signifying neurons projecting back down. So there's a very strong bidirectional connection between these 2 structures. And so our kind of working hypothesis, and you will see this figure a few times during this talk, was that really what's going on here is that you've got 2 separate pathways coming from the amygdala. One of them, which connects the amygdala to the ventrolateral frontal cortex, is really important for signaling outcome probability and learning about kind of reward probability. Whereas the connections between amygdala and orbital cortex, OFC seen here on the ventral surface of the brain, are really important for value updating and specifically signaling the identity of the outcome or the kind of reward that you might get. And so what Megan and I decided to do was to look at this interaction between amygdala and ventrolateral frontal cortex. And we also had good evidence that these types of pathways might be involved in probabilistic learning based on a set of experiments that Matthew Rushworth's group had done in Oxford. And so here they had monkeys learning in probabilistic and actually in this case just kind of deterministic situations. And what they found very nicely was that this ventrolateral area seemed to be really important and involved when monkeys were having to choose between different options and when those options reverse backs and forwards. At Mount Sinai, we've actually been doing some neuroimaging ourselves and have found something very, very similar, that when animals are playing in or learning in probabilistic reward environments, we can find very strong involvement of this kind of this ventrolateral area as seen here, and it's bilateral. And actually it seems to be even more engaged when monkeys are actually having to learn about novel probabilistic associations. But one of the pieces of information that really led us to think that there was interaction between amygdala and this ventrolateral region was part of this study that Matthew had done. And they found that there's higher functional connectivity between amygdala and ventrolateral frontal cortex during learning, especially when monkeys have to, when they don't get a reward and then decide to change their behavior. But what Megan and I were really interested in trying to understand is what's actually the function of that pathway? What's the function of the projections from amygdala to the ventrolateral frontal cortex? And so we came up with kind of two hypotheses. The first is that maybe what's happening is that when the animal makes a choice and receives a reward in a probabilistic situation, the feed-forward connections from the amygdala to the ventrolateral frontal cortex are really important for signaling that a reward, either reward or not reward, has been received. The alternative potentially is that instead, actually, maybe what those connections are for is for maintaining a trace, a kind of a memory of the choice that you make before you receive the reward feedback. And these two don't have to be mutually exclusive, but this was kind of what we started wondering, is maybe actually this is what this pathway is really important for. So to test these two different ideas, what we did is we developed a kind of a slight variation of the kind of classic probabilistic reward learning task that many of us have used in the field. So very simply, monkeys on a touchscreen would touch a central stimulus to initiate a trial. Two images would be presented on the left and right of the screen. The monkey could then select one of those, and there was a brief feedback period where they were shown the image that they'd chosen. There was then a trace interval, so an interval where there are no stimuli on the screen, and the monkey just has to remember that choice. Then feedback is given, and there's an intertrial interval. But because what we wanted to do is have within task control, what we do in this situation is we actually have two pairs of stimuli. So on randomly interleaved trials, the monkeys are presented with two pairs of images, and they have to learn which pair in those images is associated with an 80% probability of getting reward versus a 20% probability. And monkeys complete 200 trials a day, 100 trials for each pair. But what we do here is for one of those pairs of images, the animal receives the reward immediately after making their choice. But for the other pair, they don't, and they actually have to wait a second with an unfilled trace interval, so there's a delay before their feedback is delivered. And then to kind of make sure that we're not just simply changing the value of the different options in the task, we then adjust the intertrial interval to keep the temporal reward rate the same. So we did a lot of an enormous amount of kind of validation of this task, and what I'm showing you here is the monkey's performance. So for pair one, which is the control pair, versus the experimental pair where you have this trace interval. And so what you can see is that performance, the monkey, in a group of 5 animals, the animal's performance is significantly impaired as you increase the delay to the point where they learn much, much slower when there's just a 1.5 second delay between the choice that they made and the feedback that they got. And what's really nice about this, of course, is it now allows us to go test out these two different ideas about whether or not the ventrolateral frontal cortex to amygdala projections are important for just signaling reward feedback versus actually being involved in kind of maintaining a trace of the previous choice. So to get it, so then, well, that's the task side of things. What we wanted to do is probe this in a pathway-specific way. And while many of you are probably aware, you could do this using, say, disconnection lesions in non-human primates. But what we were really interested in doing is developing pathway-specific human genetic modulation methods. And so what you're seeing here is a schematic of our surgical approach, and then histology from one of our pilot animals, where we put canine-associated virus 2, which had a CRE in it, which is retrogradely transported. We injected that into the ventrolateral frontal cortex, and we made stereotactic guided injections into the amygdala in the same surgery. And what you're seeing here is labeled neurons. So when these two viruses combine, because the virus that was injected into the amygdala is under the control of CRE, what you get is neurons that project specifically to the ventrolateral frontal cortex being labeled. And that's what you're seeing here. So this is a large shot of the amygdala, and then these are the actual individual neurons themselves. So these are the neurons that only project from the amygdala to the ventrolateral frontal cortex. So with this technique in hand, we can then actually really go and probe out the role of this pathway, because what we can do now is we can feed the animals the dread activating ligand and actually alter the activity, inhibit the activity in the neurons that specifically project just from the amygdala to the ventrolateral frontal cortex. So here's the performance of our animals when they're given the vehicle. What you find is that they learn very, very quickly when they get immediate feedback, whereas they're a little bit slower when the feedback is delivered after a 1 second trace interval. And that fits our hypothesis, right? That increasing the delay has a detrimental effect on learning. But to test our two hypotheses, just to kind of go over them again, if we think that the amygdala to ventrolateral projections are really important for signaling feedback when you're having to learn in these types of environments, you would expect a deficit on both of the pairs of stimuli. However, if the amygdala to ventrolateral projections are really important just for maintaining a choice after you've made it until feedback arrives, you would expect to see a bigger deficit for this pair, where you have this trace interval between your choice and when the reward feedback's delivered. So place your bets. What we found was actually that inhibiting the projections from amygdala to ventrolateral frontal cortex produced a deficit on both pairs. So monkeys were statistically slower to learn which of the two stimuli would be associated with a higher probability of reward for both stimuli. So this really indicates to us, or it strongly indicates to us, that it's these projections that are really important for signaling reward feedback, so that you can make an association between the choice that you made and a reward that you got. And I think there's really interesting questions then of kind of wondering, okay, which parts of the brain might be involved in maintaining the choice until the reward is delivered. That could be actually the function of the ventrolateral or other parts of the brain that it's interacting with. Yep, so there's that. So yeah, this really kind of, at least to us, indicates that it's a specific projection, or at least we've now found one part of this potential network of areas that's involved in signaling reward when it's received, and you need that when you're actually having to update and learn in probabilistic environments. So that kind of, again, fits very much within this kind of part of the expected value equation for kind of learning about probabilistic reward. So again, I'm just going to quickly check that I'm still seeing you. Yep, still online. Okay, great. So with this role for amygdala to ventrolateral frontal projections in kind of reward learning kind of more heavily established, myself and another postdoc in the lab got really interested in wondering, okay, so we now know that this pathway is involved, but that doesn't tell us anything about the mechanisms that might be being engaged when monkeys are or humans are having to make choices based on the probability and the value of the different options that they're choosing on. So to get at this, myself and a postdoc, Fred Stoll, decided to really go and probe this out using electrophysiology. And so again, kind of our working idea here, based on the previous lesion work, was that maybe what's going on is that you've got much stronger kind of identity or value specific coding in the orbital frontal cortex, whereas this part of ventrolateral frontal cortex might be really important for signaling the probability that you might receive a reward. And that despite many years of research, this kind of question, at least at the level of single neurons, hadn't really been well looked into. So again, we kind of started with this kind of general idea. Maybe what's going on here is that projections, or so the amygdala and ventrolateral frontal cortex are really important for signaling outcome probability, where it's again the amygdala and orbital cortex that might be really important for signaling the identity of the different outcomes that you might be choosing on. But we also wanted to ask something else, which is that we've known for quite some time that representations, or these kind of ideas about value in the brain, are of course not stable. They change over time. I mean, in our daily lives, we might decide to eat a large meal that we really, really like the idea of. But if you, let's say you ate a hamburger, you might then decide, if someone then said, "Oh, you really like hamburgers, do you want another one?" After you've just eaten one, you might say, "No, that I don't need any more." And we've known about this type of thing. This is actually what's being assessed in the devaluation task. People like Ivan Durajo, working with Edmund Rolls, this study by Tom Pritchard's group, looking at how neurons in the brain, their activity is altered by satiety, kind of indicate, and this work by Sebastian Bure and Barry Richmond, really indicate that neurons in the frontal lobe, especially in the orbital cortex, are really tracking how values change dynamically. And so what Fred and I were really kind of got interested in is we wanted to probe how the parts of the frontal cortex would actually track these changes and these preferences that we have as they change either across days or across kind of hours or kind of seconds. And so, yeah, so again, we kind of wanted to really ask, like, how might preference or even satiety alter the encoding of outcome probability or outcome identity in these circuits? So to get at this, we designed a very, very simple task. And it's one that many of you who work in this area are probably very familiar with. Animals were shown a central fixation spot, they fixated, then 2 images would be presented to the left and right of that fixation spot, and they signaled the probability of reward for one juice and another juice. And we intermix trials, so sometimes the animals are choosing between different juices, and sometimes they're choosing between juices that are actually the same. So that's our way of checking that the animals really know the probabilities that they're choosing on. And then once those stimuli are presented, they can then make a choice and choose whichever of the 2 stimuli that they like. And what we found is that when we modeled animals' choices, we get a really strong fit of our model if we just fit the probability of the 2 different options and then the previous identity which they were choosing on. So that's nice, that fits really well. And that kind of allows us to now go on and kind of test out the way that the animals and the neurons in their brain are actually evaluating these different options. But what we also noticed, and this is important for later, is so in many sessions, monkeys' choices were well explained by this type of plot. So this basically shows you that they have an indifference between the 2 juices, and they're very strongly using the probability of the different options presented to them to guide their choices. This, again, is essentially what's being shown by this statistical estimate over here. But kind of more interesting for us was actually this wasn't always the case. And in many sessions, what we actually found was that monkeys actually had very strong preferences for one of the juices over the other, which they were choosing on. So take this scenario here, they would choose, if they would have to be offered an 80 or kind of almost 75% probability of reward for juice 1 to choose 50% of the time on juice 2. So they have a very strong preference for that juice. And that will come up later, and I'll talk to you a little bit about that in a moment. But what we now wanted to do is just start to kind of say, okay, can we find evidence of different types of encoding across ventrolateral frontal cortex, OFC, amygdala, and other parts of the brain? So we recorded in area 12, so that's ventrolateral frontal cortex, orbital cortex, area 13, 11, inferior frontal gyrus, area 44, 45, anterior insular striatum, amygdala, using these kind of wide-scale semi-chronic microdrives from grey matter research. And what this allows you to do is record day after day deep in the brain without having to remove electrodes every single day. And we can show you very, based on our histology, that we recorded, these are our recording locations in amygdala, and we could also do CT scans to check the electrode locations, confirming again that they were within the orbital cortex and ventrolateral and all the areas that I've just told you about. The other bonus of these drives is that you can record enormous numbers of neurons, at least enormous by kind of standards of kind of 20 years ago. And so you can get a really kind of a good view of what's happening in the brain. So how did neurons in the different parts of the brain we recorded from encode these different aspects of the task? So that, again, the chosen identity, so that's this kind of proxy for value, because you have to know the identity of something to kind of be able to value it, versus the probability. So if I show you the chosen identity coding first, what we found, as many others had done, was that you actually do have many neurons in orbital cortex that are encoding the specific identity of the options that the monkeys are choosing on. But we also found that type of encoding in a number of other places, so inferior frontal gyrus, ventrolateral frontal cortex, and amygdala, but less so in agranular insula and striatum. But this is just the count of neurons. This is the proportion of neurons that we recorded from that had statistically significant coding. And that's, of course, not exact. That doesn't mean that that's how the brain's signaling this. So the other way you can look at this data is to look at how much variance is explained by the neuron's firing rate. And when you do that, something slightly different comes through. Instead, what you actually find is that there's a much greater amount of variances explained by the neural activity in the orbitofrontal cortex. And again, this starts to suggest that this part of the brain is really important for signaling the identity and therefore the value of the different options available. We then, to kind of take this one step further, we trained a linear decoder on the data and then took a decoding approach to seeing how neurons in different parts of the frontal cortex would encode this part of the task. And what we found here, again, kind of backing up what we'd seen from the explained variance analysis from our encoding analysis, was again that it was the orbital cortex that had the highest decoding performance amongst all of the areas that we'd recorded from. Ventrolateral and inferior frontal gyrus were slightly lower, the amygdala was kind of intermediate as well, and then again the agranular insula and striatum were kind of very much the lowest. So at least to us, this suggested that actually there was a much better performance in the orbital cortex, indicating it had a more kind of specific role in encoding the identity of the different options that the monkeys were choosing on. We could also look at the way that probability is encoded, and here things were a little clearer. So again, what you're looking at here is the proportion, or the number of neurons that we recorded from in ventrolateral and other parts of the brain that encoded the different probabilities that monkeys were choosing on. And what you can see here quite nicely is that ventrolateral frontal cortex and inferior frontal gyrus have the highest probability of encoding this aspect of the task, and that's actually mirrored through from the explained variance analysis, again with actually the ventrolateral frontal cortex having this extremely strong representation of outcome probability when the monkeys were choosing on. And this, again, just so you're clear, this is aligned to the stimulus onset at 0, and this is the encoding of neural activity around when the stimuli are presented, when the monkeys then make their response, and then when they start to get their feedback here. Again, we can run the same decoding type approach on the data, and again, this really kind of backs up what I just showed you over here. So this is, again, just a decoding approach where you can see that the activity in ventrolateral frontal cortex and inferior frontal gyrus has the highest likelihood of decoding the probability from the activity of the neurons in that area. So this is really nice. This actually gives us this kind of separation, or at least starts to kind of indicate that there is this differential encoding between orbital cortex and ventrolateral frontal cortex for outcome probability and outcome identity and outcome value. Rather interestingly, the amygdala is kind of intermediate, and we're still trying to kind of figure out exactly why that's the case. And we're kind of, some of our analyses are kind of focused on that point right now. So yeah, just to sum up, there's this kind of difference in encoding. So for the identity of the different outcomes, OFC seems to be more kind of engaged during that portion of our task. Sorry, it's by that aspect of our task, whereas ventrolateral and in inferior frontal gyrus seems to be much more important for signaling outcome probability. But then, of course, what we wanted to understand is how do preferences impact this type of encoding. And so, to take you back to what I showed you a moment ago, what we knew is that monkeys had very strong preferences for the different juices that they were choosing on. Again, this is kind of like one of us having a preference for, say, preferring apple juice over orange juice, or grapefruit juice over, say, cranberry juice. But one of the things we wanted to understand was if these preferences were actually stable, in the same way that one of us might have a stable preference for kind of, say, wanting to drink Coke over Sprite, or Fanta over Sprite again. So what we did here is we computed the kind of a transitivity index. So let's say the animal prefers juice A over juice B and B over C. What we then wanted to check was that they would prefer A over C. To do that, we calculated this identity preference index, and what you would expect is if they are actually being transitive, that their choices and their representation should be on the kind of on this diagonal, and that's exactly what we found. So this at least starts for us to confirm that the animals did actually have stable preferences between the juices that they were choosing on, and that's really important for what I'm going to tell you about in a moment. So then we started to wonder, how would these preferences change the encoding of both juice, the identity of the juice, and also the kind of the probabilities of the juice? Because I think, as we can all appreciate, what's going on here is when the animals have these strong preferences, it's as though they're actually starting to kind of ignore or not use the probability of reward to guide their choice quite as much as they might normally do. So, of course, if we were to plot the absolute preference that monkeys have in each session and compare that against our decoding of both the chosen identity, so that's the type of juice they're choosing, and the chosen probability, what might happen, of course, is that there's absolutely no relationship, and the preference doesn't actually skew or change that decoding, kind of indicative of the representations in frontal cortex being kind of almost independent of this kind of choice bias that the animals have. But of course, that doesn't have to be the case. Instead, actually, you could have a positive relationship. So as preferences increase, you have an increase in both the probability encoding and or the identity encoding. You could have the opposite relationship, or you could, of course, have some kind of mix of those two, that actually preferences could change outcome probability encoding, but not change the outcome identity encoding. And so what we did here next is then we kind of took this approach, taking all of our sessions and running decoders on individual sessions and relating that to the this preference index that we computed for every single session. So as you can see, we had a lot of sessions because we recorded about 200 sessions from each monkey and had enough neurons to be able to run our decoder. And again, this is actually one of the reasons we were really excited to use these semi-chronic high-density drives, because you can actually get enough single neurons recorded simultaneously to be actually able to run these types of decoder. So what we found was that the monkey's absolute preference actually did modulate or change our ability to decode what the monkeys were choosing in each session. So what you're seeing here is that as preferences increased, we actually did a better job of decoding between the two different options. And we found that both in ventrolateral frontal cortex as well as orbital cortex. But I should say we also found it in almost all of the other areas that we were recorded in. Amygdala is, unfortunately, we didn't have enough simultaneously recorded neurons to get a really good sense of it. But I think you can agree with me that there's a very strong relationship between encoding of outcome identity or the chosen identity and preference in all of the areas we recorded. So it seems like this really kind of broad distributed signal. But then we wondered, okay, how would these types of preferences change probability encoding? Because of course that's probability is the aspect of the task that monkeys are using much less. And what we found here was quite interesting, that when you just take your ability to decode chosen probability and relate that to absolute preference, what you see is a negative relationship both in ventrolateral frontal cortex and orbital cortex. It is statistically significant for the striatum over here, but you should ignore that because actually it's mostly driven by a single monkey, and so we're kind of choosing to kind of, well, at least I would strongly suggest that you kind of ignore that point. But so that we found this very strong effect in both ventrolateral frontal cortex and orbital cortex. But of course, what we're doing here is we're actually looking at our ability to decode for both types of juice and not individual juices. So we kind of wondered, okay, maybe what's really going on is that while this could be driving it, you might actually have something, this might be masking a wider effect. So what we did here is we then trained two different decoders, one for juice, for the first juice, ID1, and another one for the second juice. And then what we did is we took the difference between those two. And this is, of course, our way of looking at how preferences for one of the juice might actually be skewing the representation or the kind of the probability of receiving that juice that the brain encodes. And in this situation, what we found was really interesting. Amongst all of the areas, only one of them showed this type of modulation, and that was the ventrolateral frontal cortex. And so what's happening here is that while the representation of the juice that the monkey prefers, our ability to decode that, the probability of that juice is maintained. Our ability to decode the probability of the other juice actually dies away. And this makes sense, right? Because it means that that's exactly what happens as the monkey starts to prefer one of the outcomes. But it's really interesting, it really only happens in one place that we record it from. And it starts to suggest there is actually kind of like a specific role for these areas in encoding different aspects of the expected value equation, and that those can be distorted as the animal's behavior actually starts to change. So what I've shown you here based on this experiment is that monkey choices, of course, depend on the outcome identities and probabilities. That's these two sides of the expected value equation. We can find these quite nice and clear differences between how these types of information are being encoded in the brain. Kind of backing up earlier experiments from lesions, we can find higher and a greater number of neurons encoding outcome identities, that's the outcome value in orbital cortex, whereas ventrolateral frontal cortex and inferior frontal gyrus seem to be much more important to have a high representation of encoding the probability of the different juices. I should say that this ventrolateral frontal cortex that I'm talking about here is area 12, which is on the ventral surface of the brain, and the inferior frontal gyrus is area 44, 45. And actually both of those two areas were in the lesions that I made when I was working with Betsy Murray. So that's kind of part of both parts of the ventrolateral frontal cortex. But on top of that, what we found is that when monkeys actually start to show a preference for one of the two juices, the encoding of probability is actually distorted, but very specifically, it's distorted in the ventrolateral frontal cortex. And what we think is really interesting about this is that it starts to tell you this kind of area must have a very, very specific role in encoding these types of aspects of the task, especially if this is the only place in the brain that you really start to see this change in encoding. And so while I realized I was a little bit late getting started, I'm going to tell you about one final project that's really kind of tried to kind of bring kind of an anatomical explanation to these two types of studies that I've told you about so far. So I'm just going to quickly check that you can still see me. I think you can, and you can still hear me. Okay, so this project was has been led by a very, very talented graduate student, Zach Zeisler, in the lab. And specifically, it came out of this question about that kind of stemmed from the previous study that I've showed you or talked to you about related to encoding of outcome, probability, and identity in frontal cortex. So one of the things that we noticed in these recordings was that obviously there's very, very widespread encoding of different decision signals. It's not the case that when we went to look for how neurons in different parts of the frontal cortex are encoding different aspects of our tasks, we found very high encoding in one area and absolutely no encoding in another. Instead, you find really quite kind of widely distributed brain signals for different aspects of the tasks. And I should also say that we saw this in many other parts of the brain that we recorded from. So when we use these semi-chronic kind of microdrives, you actually approach from the dorsal surface. And so as we were kind of going down to our targets in ventrolateral frontal cortex, we got to record in places like the sensory motor area, the dorsal premotor cortex, dorsolateral frontal cortex, as well as many others. And one of the things that really jumped out to myself and Fred Stoll when we were starting this experiment was that the dorsal premotor cortex, which really you might not have expected to be heavily involved in signaling outcome probability, had this extremely strong representation and an extremely early representation of outcome probability in the task. And that was really surprising to us. And we started wondering, okay, how could that be the case? I mean, this is much, much closer to kind of the kind of motor effector parts of the brain as you're trying to actually like guide choices. But clearly those signals are getting there. Outcome probability signals are arriving at dorsal premotor cortex extremely quickly. How might that be happening? And so our working hypothesis was maybe what's happening here is you've got connections from the amygdala that are actually going to the dorsal premotor cortex. And actually, to our surprise, when we went back and looked at the literature, David Amerald's group had actually shown that there's a direct projection from the basolateral amygdala to dorsal premotor cortex. Again, this is buried in kind of a number of studies back in the 1980s, but it was really interesting to us and started making us wonder, okay, well, how are these connections organized? Of course, most of us, when we think about how the brain is organized, kind of one of the easy ways of thinking about it is that single neurons have a single connection to, say, one target structure. But that doesn't have to be true. And I think it's fair to say that we already know that that's not true. Instead, it could, of course, be the case that single neurons are connected to to a number of target areas, and they're all connected, kind of intermingled. Or maybe actually what you've got is that you've got a kind of some neurons have a specific connection to one area, and then others have multiple connections. So there's kind of difference between kind of like a neuron that, say, broadcasts a signal broadly to multiple areas versus another neuron that really has a kind of a specific connection to one single area, and that provides much more kind of specific information to downstream targets. So, of course, what we were wondering is how might the amygdala neurons in monkeys connect to multiple parts of the frontal cortex? And our kind of hunch was that maybe what's really going on is that you actually do have single neurons in the amygdala that are broadcasting information quite widely, and they're doing that by bifurcating and projecting to multiple areas simultaneously. But one of the problems with studying that type of those types of projections is that none of the currently available techniques were not very good for at doing that. But recently, Tony Zador's group and a few people working with him, Eustace Kebschel and Xiaoyan Chen, have developed these really novel and really cool, really interesting approaches for getting at this problem. And one of them is called MapSeq. And very briefly, what you do is you use a virus to essentially barcode, using mRNAs, you barcode different individual neurons. Why is that useful? Well, what you can do is you can inject the virus that has these virus particles that have individual barcodes into your target area of interest. You then let the virus express, and these barcodes are expressed in the neurons and then transported down the neurons' axons to the terminals, where you can then sacrifice the animal and then read out using sequencing where those mRNA barcodes ended up. And so if you find an mRNA barcode at a distal location, and you knew that that barcode was expressed in the place that you injected the virus, then you know that there must have been a connection. And the nice thing here is that, of course, you can read out multiple target locations across the brain. So this type of approach was only optimized in mice. And so working with Zach Zeisler, this student, we decided to see if we could get it to work in non-human primates. And so based on our ideas about how decision signals and things like probability might be being signaled in the brain, we decided to use this we decided to try and pilot it in amygdala. So what we did is we injected the barcoded Symbis virus into the amygdala. We then waited a number of days, sacrificed the animal, and then sequenced tissue from these different parts of frontal cortex, including insular, entorhinal cortex, dorsal premotor cortex, cingulate, parts of striatum, and then obviously the kind of parts of ventral and medial frontal cortex that I've been telling you about so far. And what we found in 2 animals, so we managed to label about just over 3,000 neurons in the amygdala. What you're looking at here is each line is the barcode had to be found in the amygdala. So that's why this side of the plot is just completely black. And then you'll see a tick mark wherever we found a barcode above our threshold in other parts of the brain. So you can see when we collapse across this plot, you've got many more copies or many more barcodes in places like entorhinal cortex and nucleus accumbens. And if we just collapse across this matrix over here and just run the average, you get a sense of the kind of strength of projection from amygdala to different parts of the brain that we're interested in. And importantly, this strength of projection very closely matches the work that people like Helen Barbas have done previously, showing that the amygdala has very strong connectivity to nucleus accumbens, seen here, entorhinal cortex, hippocampus, agranular insula, which is this posterior part of the ventral frontal cortex, as well as other parts of the frontal lobe. So this is really important, right? If we hadn't been able to recapitulate the prior work, our technique wouldn't be working very well and we'd have to go back to the drawing board, but instead it did. So it recapitulates the kind of the bulk tracing and bulk effects that people have found. So then, of course, we want to start asking, well, how many target structures do individual neurons project to? So if you see a 0 here, that means that we couldn't detect the barcode anywhere outside of the amygdala in frontal cortex, or at least the parts of frontal cortex that we sample from. It's very possible that those neurons project to other places, such as the temporal lobe, hypothalamus, or other parts of the brain, but we didn't sample from them, so I can't say where they went. What I can tell you is that a large number of neurons had a single projection target, at least that we could detect, but many more actually had multiple projection targets. About a third had at least 2 targets, and then many more had over 3 targets as well. And so, of course, what we want to do is start to understand the structure of those projections to multiple targets. So what we're going to do here is I'm just going to concentrate on the projections from amygdala to parts of the ventral frontal cortex. So here, the agranular insula, medial OFC, lateral OFC, and the ventrolateral frontal cortex. And so the first thing we did is looked at how these projections were either specific to a single area or were kind of overlapping with many other areas. So what you're looking at here is a Venn diagram of the number of the counts of the barcodes that we found in each area that were either specific to that area. So in AI, you have 400 barcodes that were only found there in frontal cortex, versus when they were found in kind of AI as well as other parts of the ventral frontal cortex. And the point to take away from this plot here is that whereas you have this very high number of specific connections to the agranular insula, the ventrolateral frontal cortex has the least specific projections going to it, in the sense that it is much more likely, it's more than 50% likely, that the projections to the ventrolateral frontal cortex will actually, from amygdala, will be projecting to another location in ventral frontal cortex. So it's this difference between specific projections and these more widespread ones. So neurons, so the projection to ventrolateral frontal cortex is more broadcast. It actually is being spread to many other areas, whereas the projections to the agranular insula seen here are actually very specific, and they're really likely to only go to the agranular insula, at least in the ventral frontal cortex. So of course, what we'd like to do is to understand the organization of those projections a little better. And what I'm showing you here is, again, is the projections that only go to the agranular insula, and this will then plots out where else they go. So they have a very, very strong likelihood of projecting to nucleus accumbens, subcallosal anterior singular cortex as well, hippocampus and entorhinal. But of course, this plot is really difficult to look at. A much easier way, again, is to collapse across it and kind of plot these projections by area. So what you're seeing here is amygdala connections that either only go to the AI, only go to the agranular insula, medial OFC, lateral OFC, and ventrolateral frontal cortex. What you can see is there's really quite big differences in the way these neurons bifurcate to other areas. So again, if you look at the neurons that project specifically to the agranular insula, they have a very, very high probability of also projecting to the nucleus accumbens, as well as the insula, and as well as the entorhinal cortex. Whereas neurons that are projecting to the ventrolateral frontal cortex from amygdala have this really high probability of projecting to the entorhinal cortex, much less to the nucleus accumbens, kind of intermediate for the putamen, but also have a very strong probability of also projecting to the dorsal anterior singular cortex. And actually this motif, these projections that bifurcate from the amygdala to ventrolateral are really interesting to us because we really didn't expect them to have this high probability of going to also projecting to dorsal anterior singular cortex, but they do. And that really starts to tell you that there are kind of specific networks, at least of these amygdala projections, going to different parts of the frontal cortex. And it's really important to remember as well, the ventrolateral frontal cortex and dorsal anterior singular cortex are not close together. So these neurons are really bifurcating and going to very, very different parts of the brain, indicative of these kind of larger, widespread networks that might be involved in how we make decisions and choices. And so that's where my group is really, really focused now. We're really interested in trying to understand how we might be able to connect these findings about anatomy and these ideas and the fact that you actually have individual neurons that seem to be forming these kind of anatomical networks, how we might be able to link that to neurophysiology, because of course that's actually how the connectivity of neurons, at least, it doesn't completely set it, but it helps to kind of constrain how networks are functioning and how kind of physiological signals might be conveyed across those networks. And then of course what we'd like to do is relate that to actually kind of causal manipulations of function, so we can really start to understand the kind of necessary basis for the role of different parts of the brain. And with that, I think I'm going to quickly jump ahead and say that obviously I didn't do this work on my own. This was a kind of a massive team effort, and was done with many, many talented people in my lab, most notably Frederick Stoll, Megan Fredericks, and Zach Zeisler, who are the ones who really led the experiments that I've told you about today. I'd like to thank my funding sources and collaborators, both at Mount Sinai and at other locations. Thank you so much. If anyone has any questions, I'd love to hear them.